We are now moving into Chapter 9, entitled Genetically Modified Organisms, Gene Expression, Mutation, and Cloning. The chapter can be divided into these four major sections: gene expression, recombinant DNA technology, genetically modified foods, and lastly, genetically modified humans, which includes a study of stem cells. As we begin this section, we'll cover in this video lecture gene expression, which includes transcription, going from DNA to RNA, processing of the RNA, as well as translation, going from RNA into proteins. And lastly, I'll briefly cover how this whole process of gene expression is regulated. What exactly is gene expression? Well, it's going from the language of DNA into RNA, which is basically a transcription from that double-stranded helical model that we know of into single-stranded language of nucleotides, and then that gets translated into a whole new language, the language of proteins, which is just a string of amino acids. The central dogma of molecular biology is that DNA gets transcribed into RNA, which is single-stranded, and RNA gets translated into proteins, shown here as a polypeptide chain, a link of a bunch of amino acids. So again, transcription you can just think of as copying information, similar to a way that a secretary transcribes notes. Translation is going into a whole new language, going from the language of nucleotides into the language of amino acids. So what exactly is protein synthesis? Well, it's the process by which we use instructions carried on a gene to create proteins. So again, going from the information contained in your DNA such that we can ultimately make proteins. First of all, we're going to have to go from DNA into RNA. This includes understanding what exactly is a gene, which is just a sequence of DNA that encodes for a protein. And then again, that protein is just a large three-dimensional shape that's composed. DNA, by way of reminder, is double-stranded, as we've talked about. Each nucleotide contains a deoxyribose, that specific sugar lacking the oxygen. It also includes the phosphate backbone, shown here in purple, as well as one of four nitrogenous bases. Those can be either adenine, thymine, guanine, or cytosine. RNA, on the other hand, is single-stranded. It also looks similar in nature to DNA, except for the sugar, rather than being a deoxyribose, it does contain that oxygen, shown here highlighted in the white. It also contains the phosphate as well as a nitrogenous base. The only difference here is you also have adenine, cytosine, and guanine, but you do not have thymine. Instead, all thymines are replaced with a uracil. So again, we are going from DNA to RNA to protein. The DNA is double-stranded, the RNA is single-stranded, and the proteins are just a string of amino acids. So let's talk about that process of going from DNA to RNA, which we call transcription of a gene. Several sub-steps, which include initiation, elongation, and termination. So when we're transcribing a gene, you should understand a few key concepts. The RNA polymerase is able to bind to the promoter of the gene, and the promoter lies just upstream of the gene itself. RNA polymerase, once it's bound, is able to move along the gene or the DNA, and it allows for base pairing of RNA nucleotides with a complementary DNA nucleotide. This new structure is forming RNA, which we call mRNA, from messenger RNA. So at the process of initiation, RNA polymerase has to bind to the DNA, and you want it to bind before the actual gene itself. So we say that it binds in this promoter region, which is just before the gene. So RNA polymerase will bind. It's able to unwind the DNA double helix such that transcription of the actual gene itself can begin. So during the process of elongation, RNA polymerase will move down the gene and allow for various ribonucleotides to complementary base pair to the DNA, such that you are generating a complementary sequence to the template strand. Once the gene has been completely transcribed, RNA polymerase will encounter a termination signal in the DNA. This signal will allow for the dissociation of the RNA polymerase from the DNA, which allows the DNA to then reform its double helix, and the complementary mRNA will dissociate as well from the DNA and the RNA polymerase. In terms of understanding which strand the actual gene is on, we call the strand that contains the gene the coding strand. The template strand is complementary to the coding strand. And when you go to make the mRNA, it's making a complement to the template strand, such that the transcript that is made is going to look like the coding strand, with the exception that the T's have been replaced with the U's. So as shown here, the yellow complementary copy is similar to the red strand of DNA. So that covers the process of transcription, and now we will talk about how the RNA is processed now that it's been made. And this includes 3 key steps: the addition of a 5' cap, the splicing of the mRNA to remove introns, and lastly, the addition of a poly-A or a polyadenine tail. This is showing the 5' cap. This gets added on to the 5' end of the mRNA. This is the first end of the mRNA that gets synthesized, and this just gets added on and helps to prevent degradation of the mRNA that was generated. The second part of processing of the mRNA is that splicing has to occur. When the mRNA gets made, it includes both exons and introns. The introns, however, are not necessary for the formation of the protein, so those regions must get removed from the transcript that's made. And the way I remember this is that the exons ultimately get expressed into protein. So through a mechanism that I'm not going to go into detail for this course, the introns will get spliced out and you piece together the exons. Now, why advantageous? Well, you can see that from a single messenger RNA, we can generate lots of different types of proteins depending upon how we undergo that splicing mechanism. So, for instance, if we have a DNA that encodes for an RNA, and now you have a series of exons and introns, shown here as exon 1, exon 2, intron 2, etc. For the protein shown in A, all the introns, intron 1, 2, 3, and 4, were removed, and we're left with an mRNA showing 1, 2, 3, 4, and 5 exons. And that generates a particular type of protein. However, we can do different type of splicing. So as you can see in the middle protein, for protein B, what took place is that we actually spliced out intron 2, exon 3, and intron 3. And you generate a messenger RNA that includes only exons 1, 2, 4, and 5. So we've generated a completely different protein that's going to have functions that's distinct from protein A. However, what you see in protein C is that we've actually excised out exon 4, and so that gives you a whole other protein. So this is one of the ways that from a minimal number of genes, we actually have many more proteins than what the number of genes would cause one to assume that we could make. The last step of mRNA processing is that we add on a set of adenines onto the 3' end of the messenger RNA. And this also helps to prevent degradation once the messenger RNA leaves the nucleus and moves into the cytoplasm. So the function of all this processing is that it facilitates transport out of the nucleus, it protects that messenger RNA from degradation once it's in the cytoplasm, it helps to maintain stability of the mRNA for the process of translation, and the splicing variants greatly increase our genetic complexity. So that completes our discussion of RNA processing, and last I'll highlight the process of translation going from RNA into protein. This includes 4 key steps: initiation, elongation, translocation, and termination. So when we talk about translation, you should understand that this is happening in the cytoplasm. So that messenger RNA has moved out of the nucleus. It requires a set of important molecules. It includes mRNA, it includes amino acids. You need energy to drive this reaction, which comes in the form of ATP, and you need some additional helper molecules, namely ribosomes, as well as transfer RNA, and I'll explain what those do. Ribosomes are comprised of proteins as well as ribosomal RNA, and there are 2 subunits. There's a small subunit and a large subunit. When the large and small subunit come together, it forms 3 distinct sites in this huge protein RNA complex. It generates an E site, a P site, and an A site. And I'll explain what those distinct sites are more in a second. The tRNA, on the other hand, is carrying the amino acid, and it has in the bottom of this T-like structure an anticodon that is complementary to the codon carried in the mRNA. And codons are this cluster of 3 nucleotides. So how does this process work? First of all, the ribosomes are going to attach to the mRNA at the 5' end. The ribosomes are going to facilitate the docking of a tRNA anticodon that's complementary to the codon in the mRNA. When 2 tRNAs are adjacent to one another, a bond is formed between the amino acids at the other end of the tRNA molecule. A peptide chain is formed by linking together these amino acids. So the way that this looks like is you will have the DNA transcribed into mRNA in the nucleus that is then exported out of the nucleus into the cytoplasm. We then have our single-stranded mRNA floating in the cytoplasm. The ribosomes come together, shown here in the light gray structure. The tRNAs are then going to bind to the mRNA, specifically by the complementary anticodon that's complementary to the codon in the mRNA. The other end of the tRNA is going to have the amino acids, and as each additional tRNA comes into that structure, this growing polypeptide chain gets transferred on to the new tRNA until we ultimately hit a stop codon, which indicates that the complete protein has been synthesized. So how do we know which anticodon is complementary to which codon and how that specifies for a particular amino acid? Well, we have a genetic code. So this genetic code allows for a specific codon to code for a specific amino acid. Since the codons are comprised of 3 nucleotides, and we have 4 different possible nucleotides, this gives us 64 possible combinations. We actually have 61 codons, which actually code for specific amino acids, and then we have 3 codons, which are functioning as stop codons. There is some redundancy in the genetic code. We know that we have only 20 amino acids, but as described here, we could have coding for up to 64 amino acids, but we don't need that. And the genetic code is universal. If you're talking about this code in bacteria versus in humans, the same codon encodes for a particular amino acid. What does the code actually look like? You can see here what we're talking about when it says first base, second base, and third base. This is all in reference to the mRNA, not in reference to the anticodon. But the first base, if we're looking at a mRNA that has a codon of UUU, if you look at the upper left-hand corner of this genetic code, you can see that that encodes for the amino acid phenylalanine. There's three very important stop codons as well as a start codon. All messages, once they begin to be translated, they always start with an AUG, which is a methionine. So all proteins begin with this methionine. And the way I remember this is that school always starts in August, AUG. So that's true for all proteins. We have three stop codons, which are also universal for whether we're talking about prokaryotes or eukaryotes. These are UAA, UAG, or UGA. My way of remembering this is you are all right, you are good, or you go away. And somehow that has stuck with me since high school. So use what you need to remember these important codons. So please take a moment here and try to explain gene expression to a family member, a colleague, your pet, anyone willing to listen. So that summarizes this section of the talk on translation, which is initiation, elongation, translocation, and termination. I've also included in the module several videos that will be very helpful for you to visually see what's going on in these distinct processes. So an additional question to you is, do you think all RNAs are translated into proteins? Only mRNA is what's actually translated into proteins. As I mentioned, we have things such as tRNAs, which are transfer RNAs. These carry amino acids to ribosomes, so these are not translated into proteins. We also have ribosomal RNA, which makes up those ribosomes, along with proteins, and these are not translated either. Another question for you is, since prokaryotes, bacteria, do not have organelles other than ribosomes, how is the RNA actually processed? Well, they have no 5' cap, they have no splicing, and they have no poly A tail. Their system of gene expression is greatly simplified. Also, transcription and translation are coupled and are happening simultaneously. So it looks like the following. Here we have a prokaryotic cell on the left-hand side. Once that mRNA is beginning to be transcribed, it's almost immediately undergoing translation as well. And here you can see three different sets of ribosomes on this mRNA, and the polypeptide chain is being made as the mRNA is being transcribed. Shown on the right is the eukaryotic cell, which we've discussed extensively already. So how do we actually regulate this whole process? We know that each cell in your body has the exact same DNA, but each cell only expresses a small subset of those genes. So if we're talking about nerve cells and muscle cells, they have the same genes, but they have vastly different functions, and this is due to the fact that they express genes at different times. So it's really this turning on and off of genes that allows you to have our specific functions. So here again, these are nerve cells and muscle cells taken from the same organism. They have the exact same genetic components. However, because they've turned on and off genes at different times, you get this highly differentiated phenotype. So what regulates this process of gene expression? To a large extent, it's the binding of the RNA polymerase upstream of that gene that's mediating a lot of this regulation. So how does RNA polymerase even know where to bind? Well, there's specific sequences in the promoter that target the RNA polymerase to bind in a specific location. So here we have a series of promoters from different strains of E. coli. And as you can see, there are regions that are homologous from one strain to the next. And these are all indicating to the RNA polymerase to bind there, which is just upstream of the gene. Again, as I've said, essential sequences in the promoter line up RNA polymerase, so it starts at position 1. And position 1 is not necessarily your ATG or your AUG once it gets translated or transcribed into RNA. There can be a lot of untranslated RNA before translation ends. And what you'll see is that although these regions have high homology, they're quite similar to one another. There are some slight differences, and these differences mediate the ability of RNA polymerase to bind more strongly or less strongly to the promoter, and how strongly RNA binds is going to determine how much message is ultimately made. Another way that transcription is regulated in prokaryotes is they use things such as repressors, and a repressor can bind into the promoter region of a gene as well, and this will inhibit the ability of RNA polymerase to bind there. If you compare bacteria to eukaryotes, such as a simple eukaryote like yeast or humans, you can see that the promoter region, shown here in purple, is just upstream of the gene. However, the regulatory sequences for bacteria are usually adjacent to the promoter. In yeast, you can see there's a lot more variability in terms of what's regulating the ability of RNA polymerase to bind to that promoter. And in humans, you can see that it's much, much more complex. So for eukaryotes, it is important for the RNA polymerase to bind, but what determines whether or not RNA polymerase can bind to the promoter is based on a whole series of proteins called transcription factors. So for this eukaryotic promoter, you can see that RNA polymerase is going to bind into the DNA. This is theoretically just upstream of the gene, but all these other colored circles are indicating transcription factors that are going to help stabilize the binding of RNA polymerase to the promoter. You can also have enhancers, which are far from the promoter region, and based upon the way that the DNA is folded in on itself, these enhancers allow for the binding of other transcription factors, such as activators, that further help to stabilize the binding of RNA polymerase, ultimately leading to high levels of gene expression. You can also have silencer regions in which, rather than it being an activator, it's actually a repressor, and the repressor can inhibit the stabilization of that RNA polymerase and ultimately turning off gene expression, as I've indicated here in this X. So as I've stated, for eukaryotes, they use other proteins such as activators to help regulate gene expression, and these activator proteins can bind far away from the actual promoter region itself. So how do we regulate gene expression? We can regulate gene expression by allowing the chromosomes to be condensed or uncondensed. If they're fully condensed, the RNA polymerase is going to have a hard time binding into the promoter region of the DNA. As that chromosome relaxes, it'll open up the DNA and allow for RNA polymerase and other transcription factors to bind. We can also regulate gene expression by degrading mRNA. If there's high degradation activity, then we won't actually be able to make much protein from that mRNA. We can also regulate the process of translation by slowing down the ability of the mRNA to bind to the ribosome. Another means of regulation is protein degradation. So proteases will come and degrade the protein once it's made, and this can lead to decreased protein activity. So in summary, prokaryotes are able to regulate their genes to make immediate changes in response to their environment, and these regulatory regions are usually adjacent to the promoter regions and are in close proximity to the set of genes that they regulate. Higher eukaryotes, such as ourselves, we regulate by using combinatorial approaches, so we can have a whole set of transcription factors that are modifying the ability of RNA polymerase to bind more strongly or less strongly or not at all, and this all can regulate the amount of mRNA that ultimately is made. We utilize things such as enhancers, which are distant regions of DNA that can loop back to the promoter and affect RNA polymerase's ability to bind. The main takeaway is that eukaryotic regulation is vastly more complex than what we see in prokaryotes. That concludes the first part of Chapter 9, which covered gene expression. We talked about transcription, going from DNA to RNA, how the RNA is actually processed, and also the end step of translation, going from RNA to protein. Lastly, we also highlighted some of the many ways that gene expression is actually regulated. Thanks so much.